Hello, everyone, and welcome to Precision Biosciences' first virtual symposium. My name is Cassie Gorsuch, and I am the VP of Gene Therapy here at Precision. Today, it is my pleasure to be joined by two incredible key opinion leaders to discuss an unmet patient need as it relates to chronic hepatitis B, a leading cause of morbidity in the US and death globally. Today, we'll go over the current treatment landscape, a little bit about the virus that underlies chronic hepatitis B, the need for better curative options, and then touch on Precision Biosciences' PBGENE-HBV program utilizing our ARCUS gene editing technology, and think a little bit about what our phase 1 clinical trial design might look like. With me today is Dr. Mark Sulkowski, Chief of the Division of Infectious Diseases at the Johns Hopkins Bayview Medical Center, and Dr. Jeffrey DeShaco, Emeritus Professor of Medicine at the University College London School of Medicine and a hepatologist consultant at King's College Hospital. Thank you both so much for being with us today.
Thank you for the invitation.
Thank you. Thrilled to be here.
So first, let's start with sort of a big picture of chronic hepatitis B. Dr. DeShaco, for anyone who may be joining us who is not familiar with chronic hepatitis B, could you give us a little overview about what hepatitis B is and its effects globally, please?
Thank you, Cassie. Hepatitis B virus infection is caused by the hepatitis B virus. It's a DNA virus. It's actually an RNA virus gone wrong. There's an important global prevalence of hepatitis B virus, which is not declining, and the morbidity of hepatitis B virus infection is actually increasing. There are thought to be over 250 million people living with hepatitis B, and the vast majority at the moment are unaware of their diagnosis. And again, even amongst those diagnosed, only a minority are being treated. So it's an important global health problem which has been neglected. Times are changing. There's a new emphasis on combating hepatitis B at the present time, but it is important to understand the global epidemiology and morbidity caused by the hepatitis B virus infection.
Thank you, Dr. DeShaco, for sharing your perspective on really how this virus impacts on a global scale. Dr. Sulkowski, could you please tell us a little bit about how this virus looks specifically in the US?
Sure. In many ways, the United States reflects the rest of the world. We have about 1.5 million people living with chronic hepatitis B. Many of those people moved to the United States from other parts of the world where the virus is more common. That's about 75% of the people with Hep B in the US. Most of these people, the 1.5 million, are really not diagnosed or in care. In fact, we estimate that only about 300,000 are currently being treated. So the consequence of that, a high disease burden, very minimal diagnosis and treatment, means we have high rates of liver-related complications, including hepatocellular carcinoma. So a major cause of morbidity and mortality in the US.
Thank you both for painting that picture of what this really looks like on a global scale, as well as particularly within the US. Dr. DeShaco, you touched on sort of the underlying biology of chronic hepatitis B and that it's caused by the hepatitis B virus. Recognizing that there's probably quite a diverse audience with us today, maybe we can keep this a little bit high level, but let's talk a little bit about the viral life cycle and how this virus really leads to chronic hepatitis B.
I guess the first thing to say is, for some reason, individuals infected very early in life by mother-to-child transmission or infected in the neonatal period seem not to mount an appropriate immune response to Hep B, and childhood acquisition is the most important cause of incident chronic infection at present. For some reason, adults are less likely to develop chronic infection. They may develop an acute infection and clear it, although some adults may go on to chronic hepatitis B. The replication cycle of hepatitis B is relatively well understood, and briefly, upon entry into the cell, the relaxed circular DNA is converted by cellular enzymes into the CCC DNA. That's better known as a mini chromosome because it is an episomal mini chromosome responsible for subsequent replication of the pregenomic RNA, reverse transcription of the pregenomic RNA into minus and plus strand synthesis, and assembly of the viral proteins into the virion and subsequent spreading. There are a number of targets that have been identified within that. It's important to mention that one minor but critical aspect of the life cycle of Hep B is the formation of double-stranded linear DNA, which integrates at random sites into the host chromosome at breaks. Although those are linearized, they don't take part in the replication cycle of hepatitis B. If properly sited after the promoter, there will be continued production of large, middle, and small surface antigen. And that antigen excess over years, we think, is important in tolerizing the infant, but also exhausting the T and B cell response to Hep B, thus impairing the likelihood of clearance of Hep B. The integrants are also critical, probably, for the oncogenesis of hepatitis B virus.
Yeah, thank you for walking us through that, Dr. DeShaco. And Dr. Sulkowski, based on what Dr. DeShaco just said, there's really two forms of viral reservoirs that exist in infected hepatocytes, the cccDNA and the integrated HBV DNA. Can you speak a little bit to the importance of both of those two viral sources?
Sure, and that was a fantastic overview. I think when we think about the active infection, this is really the cccDNA. This is driving the production of new virions that will infect other hepatocytes, keeping that infection going in that individual, but they're also going to be potentially spread to other people, transmitted through mother-to-child transmission, through sexual transmission, or bloodborne transmission. So the cccDNA drives the infection, but this integrated DNA really perpetuates the production of surface antigen, which may serve as immune tolerance, but also drives potentially hepatocellular carcinoma and pathogenesis of clonal expansion of hepatocytes. So both are critical roles in terms of the infection itself.
Thank you. And I think, Dr. DeShaco, you touched on the idea that there have been a number of different therapeutic strategies aimed at different parts of this viral life cycle, and we'll get to that in just a moment. I want to first spend a couple minutes talking about these patients. And, at Precision, we always try to put the patient first as we're thinking about developing our therapeutic approach. And so I want to spend a couple minutes talking about the burden and the impact that chronic Hep B has on people on a personal level. And so, Dr. Sulkowski, could you tell us a little bit about how this virus really impacts people on a day-to-day basis?
Sure. When I meet a person who's newly diagnosed hepatitis B, there's really two major concerns that they share with me. One is about their personal health. Will they develop liver disease? Will they develop liver cancer? And can this be cured? These are the thoughts going through their head. And they carry that with them as they go through their lives, this idea that there may be cancer forming in their liver. The second element that people share is concern about transmission. How did I get this infection, and can I spread it to others around me, particularly women of childbearing potential, concern about mother-to-child transmission. So there is a tremendous amount of concern and anxiety people have, both with their individual health as well as transmission, and certainly a lot of stigma that they carry. This is not something they talk about with people outside their immediate family.
Thank you. And Dr. DeShaco, maybe you could share a little bit more about that stigma that patients face and what sort of impact that can have globally.
I would just endorse what Mark has said. I think hepatologists have probably underestimated the stigma felt by patients living with hepatitis B. We're also aware that the level of activism amongst patients with hepatitis B is much lower than patients with HIV, for example. And there are a variety of reasons for that. It's often more prevalent in minorities, amongst immigrants, refugees, etc. And they've just not had the powerful voice of the HIV community. That's changing. And I think it's up to us to address issues like stigma in many walks of life, occupational stigma in the military, immigration, visas, work permits, the ability to move from country to country, etc., transmission that Mark has mentioned. We have a lot of work to do in this area, and I don't think it's been addressed well up till now. But we are aware, I think, of a growing patient voice and would support and endorse that. It is important.
Thank you for sharing that with us. It sounds like there's really a lot of unmet need and a lot of activism needed in this space to really promote good options for patients. And so let's talk about what that looks like in terms of good options for patients. Dr. Sulkowski, you mentioned when you sit down with a patient newly diagnosed, what does that conversation look like from the perspective of treatment options and curability of something like this?
Sure, and certainly, as I mentioned, every patient I've talked to with hepatitis B wants to be cured. In fact, I often hear, well, there's a cure for hepatitis C. Why is there not a cure for hep B? And actually, Jeff shared exactly why there's not an easy cure. This is a more complicated virus. So I actually do talk to patients about the virus itself, its life cycle, and describe the current therapies, the nucleotide analogs that work to inhibit the polymerase, which reverse transcribes the RNA back to DNA in that viral life cycle. These are therapies that are safe, tolerable once daily. They can suppress viral replication, leading to less liver disease and potentially reducing the risk of liver cancer. But it is important that the patients understand that these are not likely to lead to a cure, either a functional or a sterilizing cure. We'll talk more about those.
Yeah, thank you for taking us there in terms of what that functional cure or sterilizing cure is. And maybe I'll turn it over to Dr. DeShaco to really describe what is the goal from a treating physician's perspective, from a therapeutic field perspective in terms of goals towards cures.
We've used interferon quite widely in the past, but it's much less commonly used now because of toxicity and the lack of efficacy in large numbers of individuals. So the mainstay of treatment are nucleoside analogs. They chain terminators. They affect a late stage of that replicating cycle. They affect minor strand and plus strand synthesis, but they have no direct effect on cccDNA, and they have no direct effect on integrants, only to the extent that they reduce replication, but they will not eradicate those integrants. And so they are important drugs. They will effectively suppress hep B replication, but they're used as maintenance, suppressive, often lifelong therapy. And some of our patients have been on nucleoside analogs for many years. The problem is, as you'd expect, because of the lack of a direct effect on cccDNA and integrated viral genomes, the majority of patients treated with nucleoside analogs remain HBsAg positive. That stigma persists, and that probably represents an antigen excess impairing the immune response. So we need to think laterally about curative therapies. There's a lot of effort in this area. Curative therapies are currently being defined as clearance of HBsAg using a sensitive, accepted laboratory assay, 6 months after stopping treatment, together, of course, with reduction or undetectable HBV DNA. So it represents a finite treatment of period, and after treatment cessation, individuals will be HBsAg negative, no detectable HBsAg in serum. And that will mean targeting both integrated and cccDNA to a better extent than we are currently.
Thank you. Yeah, I think that really provides a nice overview of what the current standard of care is with nucleoside analogs. And really, I think while it sounds like these are safe, well-tolerated, effective drugs, they are not offering that functional cure to patients. So what else is going on in the space? And, as you think about new emerging therapeutics, what are your thoughts towards achieving that goal of functional cure? And Dr. Sulkowski, I'll start with you, please.
Sure. Well, I think the first thing I'll say is that we are seeing a number of different therapeutic targets being identified and drugs being tested in human trials. That's really important that we see these advances. Broadly speaking, they can fall into two categories. There's ones targeting the viral life cycle. We know that the current therapies, nucleotide analogs, work on the polymerase, but there are potentially other targets. And we've seen oral drugs targeting capsid assembly entering human trials, and in some cases, suppressing viral replication. And these look like they may have an option as antivirals. There are also drugs that are targeting the translation of the pregenomic RNA, that is the viral RNA that gets translated into proteins like the surface antigen. We're seeing both siRNAs as well as antisense oligonucleotides, including one that's made it into phase 3 clinical trials. Now, so far, these antiviral approaches have had relatively modest effects in terms of their ability to deliver that S antigen loss or functional cure. So that leads me to the second broad category, which is immunotherapies, that is trying to harness the immune system to control the virus, and perhaps using these in combination therapies. That's a 1-2 punch where we knock the virus down and then boost the immune system using strategies ranging from checkpoint inhibitors to therapeutic vaccines. So all these things are being tested with some success, but relatively modest success to this point in time.
And Dr. DeShaco, as you look at these new emerging therapeutic options, and this idea of potentially combining, an antiviral with an immune modulator, what are your thoughts in terms of how that might lead to options for patients and progress towards that functional cure?
Mark's given a good summary of the state of the field. I think it's fair to say at the moment that we can achieve with small interfering RNAs and antisense oligonucleotides quite good knockdown of HBsAg, typically around 2 log, and around 60 to 70% of patients on treatment will lower their HBsAg concentrations to quite low levels, around 100 international unit. But far fewer patients are clearing the virus and achieving functional cure. And that's probably, to all intents and purposes, only being seen in patients who have low baseline HBsAg concentrations. So the current phase 3 trial with the ASO, as Marcus mentioned, is actually including patients with HBsAg concentrations below 3,000, which is a relatively low HBsAg concentration. So we've got much work to do. I think the strategy of knockdown of HBsAg leading to restoration of the dysfunctional immune response is really only being seen in patients with low baseline HBsAg. In some way, they are primed. And I to some extent, it's disappointing. I really think the immune system with acquisition in the neonatal period is markedly dysfunctional. People who've studied T and B cell function show extensive exhaustion, immunologic scarring. The cells are often resident in the liver and not in the periphery, so it's very hard to study in peripheral blood. It looks as though those cells have been imprinted away from hepatitis B. And so the strategy of immunomodulation following knockdown of HBsAg is not the final answer. It's time, I think, for that reason to start thinking laterally of other much more direct approaches, targeting cccDNA and directly targeting HBsAg transcription from integrated viral genomes in the hope that that will lead to greater rates of functional cure. But we have reached that point.
Yeah, that's a great segue, I think, to take us into the precision approach towards a functional cure for hepatitis B. So at Precision, we are developing our PBGENE-HBV program, which is a lipid nanoparticle that delivers an ARCUS nuclease, which is a gene editing nuclease that specifically recognizes a target sequence in the HBV DNA that's present in both cccDNA and integrated HBV DNA. And so the goal here is to cut cccDNA, which will lead to elimination of cccDNA, and cut integrated HBV DNA in order to inactivate and turn off any transcription from integrated HBV DNA. And our goal at Precision is really this two-pronged approach at cccDNA and integrated HBV DNA in the hopes of achieving a functional cure. And you can see we've demonstrated from my perspective, a really robust preclinical data package showing up to 99% viral DNA elimination in non-human primates. And really exciting from our presentation at the liver meeting back in November, the ability to durably inactivate integrated HBV DNA in a transgenic mouse model. And so both of these pieces of data were shared back in November, and we continue to build out a very thorough and robust efficacy and safety package to continue progressing our PBGENE-HBV product. But I'd like to take a minute and ask Dr. DeShaco, you mentioned that the time has come for a new approach. What are your general thoughts about a gene editing strategy for hepatitis B?
I think it's an exciting approach. It's clearly a departure. Safety will be paramount. Globally, chronic hepatitis B occurs in a generation who've missed hepatitis B vaccination, but there are many parts of the world where individuals who would qualify for treatment and cure would be in their 30s and 40s. So safety is paramount. The principle would be to treat hepatitis B earlier, before integration events lead to an unstoppable oncogenic event and before the onset of cirrhosis. We will have to ensure safety. And I know that Precision Biosciences have been spending many years evaluating the safety of the gene editing approach. At the same time, the idea of targeting cccDNA at the same time as targeting integrated viral genomes is an attractive proposition. So I emphasize safety. It's a new world, but the time has come, I think, to look at gene editing prospects for the management and curative treatment of hepatitis B.
And Dr. Slakowski, when you look at this approach, what makes you really excited about an editing strategy like this?
Sure. Well, I think the excitement comes from the fact that it's really getting to the heart of the problem. It is targeting the viral infection that the other therapeutic strategies, both the ones we're using in clinic today and those that I talked about in development, can't get at. They're directly targeting the cccDNA, which is the pathway to make new virions and perpetuate the infection, and even the pathway for viral reactivation and people who achieve functional cure years later. By targeting the heart of the problem, we're really eliminating cccDNA. And then if we turn to the integrated DNA, the concern is that this may be driving oncogenesis, that is the development of hepatocellular carcinoma. And if we can target that, that may reduce the risk over time. So it really gets to the problem, that is the virus and the viral DNA.
Cassie, I could interject
Please,
for a
go
moment.
ahead.
I should mention as well that we have a readily available biomarker that will demonstrate target engagement, and that's the decline in HBsAg. It really couldn't be simpler. So we will know if we are effective at reducing HBsAg, and we'll be able to understand with time the quantities of HBsAg being reduced from cccDNA and integrated viral genomes. But we have a readily available biomarkers to look at the kinetics of the response, and I should mention that.
Yeah, that's a great segue as we think about biomarkers and how we will know if this is working in patients. Maybe we can spend a little time talking about this first-in-human trial design, and I'd love to get your perspective on what types of endpoints we should be looking for, both on the safety side and on the efficacy side. And Dr. DeShaco, you mentioned, S antigen as a really robust efficacy endpoint. So maybe I'll start with you in terms of what are your expectations? What would you like to see in terms of endpoints for this phase 1 design?
Thanks, Cassie. In the broadest term, there are two major categories of patients: E-positive patients with high and less high levels of replication and varying degrees of activity, and E-negative patients where the levels of activity can vary. Some of these patients can have quite low levels of replication. So we've got good biomarkers that distinguish that pattern. I'd like to see E-positive patients included, and in those patients, we would want to see a reduction in Hep B DNA if they're not on a nucleoside analog. Importantly, hepatitis B RNA, because that's the major transcript from cccDNA, quantitative E and quantitative S. In the E-negative category of patients, you might not see as much hepatitis B RNA. It can be difficult to detect if most of the S has been derived from the integrated viral genomes, but I would certainly want to measure hepatitis B RNA if detectable at baseline, correlated antigen, and of course quantitative surface antigen together with other markers like the transaminases, to show that we're improving liver function, and perhaps Mark can amplify how we use transaminases in these trials.
Yeah, please, Dr. Slokowski, can you share your thoughts in terms of transaminase elevations, expectations around that, as well as your thoughts on patient enrollment criteria?
Sure. I think Jeff has made this point. I want to emphasize it, that we really want to make sure that we are targeting safety of this approach, this gene editing approach. And that's going to be selecting individuals who are actively infected. We want to be able to measure markers of viral replication and the decline with gene editing treatment. But we also want to make sure that they have minimal liver disease, normal liver enzymes, ideally entering or close to normal, entering the therapy. And then we need to monitor closely. I think we've seen liver enzyme elevations with a number of different agents being developed, all the way back to interferon, where liver enzyme elevations were actually seen as a marker of immune response. But we need to monitor closely with stopping criteria such that if liver enzymes increase, we can stop the intervention and monitor patients. I think we know how to do this. We've been working in this liver space for some time, but the key will be safety and patient selection as we move forward.
And as we think about, this approach, this gene editing approach, where the effect is very rapid, we expect that we would engage cccDNA and integrated HBV DNA very quickly after the LNP is administered, so the effect would be very fast. Our preclinical data really suggests that the S antigen decline happens rapidly, that any transaminase elevations are moderate, well-controlled, and very transient. And so I think both of those, from the efficacy and safety perspective, bode well as we think about moving into this phase 1 trial. Dr. Slokowski, given that information from our preclinical data, what are your expectations around what you might see and how you would manage that, as a physician, in terms of the ALT elevations potentially, or any sort of S antigen decline? If you see that, from the permanence of this approach, what would your expectations be on that changing?
Sure. The ideal scenario, we enroll participants, they're treated with the ARCUS gene editing. We see a decline in S antigen and other biomarkers. Now, at the same time, we might see elevations in liver enzymes. And in general, we're going to measure those as a fold increase over baseline. And we're really quite comfortable with modest elevations in liver enzymes, and that wouldn't lead me to change my approach to that patient as long as they're clinically well. I think what we want to see as we finish the treatment and as we observe patients, ideally we'd see the persistence of no viral markers, that is, achieving functional cure. Of course, with this approach, you have to ask the question, is it functional, or perhaps is it sterilizing cure? But we want to see that persist over time, not just over 6 months, but even longer.
Yeah, and I think our expectation is really the upside of a gene editing approach is the permanence of it. And so if you've eliminated the cccDNA and you've inactivated the integrated HBV DNA, when you see those viral biomarkers decline, there's really no reason to see them rebound. Dr. DeShaco, do you agree with that sort of thinking of how we're considering the durability of this approach?
Absolutely. And I just take Mark's point. It's correct. We've become accepting of ALT increases if things are going in the right direction, if that's associated with the knockdown of either viral replication or, as we've seen with some of the ASOs, for example, a reduction in HBsAg. And we would have other markers to ensure safety, like the prothrombin and bilirubin. But yes, the permanence of that approach, I think, is key. We would not want to see and wouldn't expect to see if patients become undetectable on treatment. The hope is that they would remain undetectable off treatment. If cccDNA is inactivated and eliminated, we're not likely to see reactivation from cccDNA. And we could study that again with sensitive biomarkers. But the hope is that this indeed would represent permanence and a cure.
I think that's a really exciting and hopeful note to start to bring this to an end, I'd like to take a moment to thank both of you. And it's really been a privilege and an absolute pleasure talking with you today. I'd like to give each of you an opportunity just to share any concluding thoughts that you have. So Dr. Sulkowski, we'll start with you, please.
Sure. Well, it's been a pleasure to talk with both of you. I've really enjoyed the discussion. I think this is really an important topic. I think when we look at the burden of hepatitis B around the world and in the United States, this is a silent epidemic that is taking a toll on people every day. So we need to continue along every aspect of the control of this epidemic, that is vaccination, diagnosis, and treatment. But these cures can't be underestimated. It's critical that we find a way to eliminate hepatitis B from individual patients, leading to both functional and what is really exciting about this approach is a sterilizing cure. So I'm excited to see the next several years as we move forward.
Thank you, Dr. Sulkowski. And Dr. Dusheko, I'd like to turn it over to you, please.
Hepatitis B is an important disease. I've seen much morbidity on 3 continents from the disease in my training and career. I think that the public health improvements in hepatitis B will follow the science. So the hope is that we can develop high rates of curative treatment and public health measures will follow. There'll be a spur to, as we did for hepatitis C, find patients, treat them, cure them, and reduce the ongoing morbidity from chronic hepatitis B.
Well, again, I'd like to thank you both for joining us today. It's been an important conversation, really enlightening to hear, the patient perspective first and foremost, and then really talk about how we think about bringing PBGENE-HBV to those patients. So thank you again for your time. I'd like to take a moment to really describe, from the precision perspective, where we are today and where we're headed with this PBGENE-HBV program. So we continue to be on track for a CTA and/or IND filing later this year and hope to really start enrolling our clinical trial very soon after that. We are excited to share that we will be sharing some important safety data from our preclinical safety package at the European Association for the Study of Liver meeting in June. And we're really committed, excited to continue this momentum for our PBGENE-HBV program into the second half of this year. So with that, I'd like to thank all of you for joining us. We're excited to bring this potential curative treatment option to patients together. So thank you all.